deep sea hydrothermal vent. So in this kind of environment, it has a lot of sulfur. So especially the sulfur element. So basically, we put this sample in the basal medium and supplement with sulfur element, and then we continue this cultivation. So you can see after that, we got this very special archaea. It has a very interesting morphology. You can see it has five fingers. It's like very like a baby cute foot. So actually, for this kind of bubbles, it is a vesicle. So what is something in this vesicle? So we did some observation. You can say in the picture. So this vesicle, it contains a lot of sulfur. We call that a vesicle cluster. So beside that, it has some nanotube. You can see this nanotube, it can form between the cells, different cells. So it has a connection between different cells. So what in the nanotube? It's also the vesicle. You can see this round things, this vesicle. Actually, within this vesicle, it has the sulfur. So that means they use this kind of vesicle of the vesicle cluster nanotube to transport this sulfur from outside to inside. So this is a very typical example for this cultivation of deep sea archaea. And the second one, we did some cultivation of deep sea microorganism based on the low temperature. So we got something from the Arctic areas. So you can see this bacteria, it's very special. So in the 4-degree, it has the best growth. You can see after maybe 40 hours growth, it can go to about 1 OD 600. But for the 16-degree or 20-degree, it has even less biomass. And also it has a very, very interesting morphology. You can see like a very special insect. It has a lot of things around the cells. And also between the cells, it has also the nanotube. You can see there are a lot of nanotube between the cells. And also if you culture this deep sea bacteria in different temperatures, it has different colors. You can see for the 4-degree, 16-degree, 28-degree, it's a totally different morphology and even the colors. And also for the next one, we did some cultivation based on the microbial interaction. You can see we got this very special morphology. For this guy, it is a bacteria. It's a new phylum. We call that OP1. It has 2 arms, 1 head, and for the round of things, it is archaea. It is the archaea cells. And if you don't see the details, I can show you a very interesting picture. You can see this is the OP1 bacteria. This is the archaea. It has a very obvious interaction. So if you can see the detail, I can show you the detail one. You can see this is a very, very special interaction between the bacteria and archaea. Actually, right now we did some details. We know why these two guys need to stay together. What is doing for this archaea, for this bacteria, and also what is doing for this bacteria, for the archaea. Actually, they have some nutrition connection for the archaea and the bacteria. You can see the clear things. So actually, for this strategy, we got a lot of very special bacteria or archaea from the deep sea. And for the next one, we got some deep sea bacteria. We call that PVC bacteria. So they have very special eukaryotic characteristics. You can see we cut the cells into different slides. You can see it has something very special like the nuclear for the eukaryotic cells. And also for the cell division, it can keep this guy inside of the cells. And even after this cell division, it has 2 different, like in the first round, we think, oh, maybe it's eukaryotic cells because it has something like the nuclear. So what is that in these cells? So after that, we did this very interesting. We cut the cells into maybe 7 different slides, and then we did the reconstruction. And then you can see. So if we do this reconstruction, you can see after that, what do we see? Okay, it's it's very interesting because when we finish this reconstruction, you can see for this in the first round we saw that maybe it's nuclear, but it's not. So what do you think that what is like? Yes, it is like cucumber, right? So it's like a cucumber. So if we search in the website, it's very like the eukaryotic mitochondria. You can see the formation, the morphology is very like. Actually, right now, so we are going to do some details. We maybe in the near future, I can get the results because we got a lot of evidence. We show that actually in these cells, it already evolved something about the mitochondria, like the eukaryotic cells. So that's one very interesting thing. And for the second part, I'm going to give some examples of the deep sea microorganism. Why we use this complex like the polysaccharide or the proteins or the nucleic acid acids to do this enrichment. For the first one, I'm going to give you samples about the deep sea microorganism use the polysaccharide, the alkene, the nucleic acid to do this enrichment. So for the first one, we use the nucleic acid from the E. coli. So for the enrichment, you say we put these cold sediments together with the basal medium supplemented with the E. coli genomic DNA and do this enrichment culture. After several rounds purification, we got a very special deep sea bacteria. We call that tenorchils. What is tenorchils? Tenorchils, it is short of the cell wall. It has no cell wall, and it has a very special trait because it has the nucleus DNA. Like it has, it can totally cut the E. coli DNA into different pieces. So in the laboratory condition, we can see the results. And also, very cool thing, we can put this deep sea bacteria back to the deep sea cold ship. You can see the white one, it is the shrimp. For this one is the oyster. So that means we can culture this bacteria in the laboratory, and also we can do the in vivo, the in vitro, oh no, in situ experiments in the deep sea. So I think this is very important for us to do the detailed exploration in the deep sea in the future. So because this guy, it has a very small genome, it's less than 2 MB. You know, for the E. coli, it has about 4.5 to 5 MB. For this guy, it has no cell wall, and also the cell genome size is very small. It's less than 2 MB. So how to utilize the nucleic acid with such a small genome? So we did some detailed study. We found actually for this tenorchils bacteria, it can produce some bacteriophage. So this, you can see clear for this virus. So we sequence this virus. In the genome of this virus, we found some, you can see the red arrows. It has told about the nucleic acid degradation associated protein. That means actually for this bacteriophages, they can help the host cells, that is the tenorchils, to utilize the nucleic acid. So it's very interesting because it has a very small genome. It cannot utilize this nucleic acid, but it can recruit some bacteriophage to help it to degradation of the utilization of this nucleic acid. And so we give this Hugel plasma because it has both the characteristics from the tenorchils and the firmicutes. And it's complex. So we use this guy to name this bacteria Hugel plasma because it comes from the classic of mountains and seas. It's a very, very famous book from China because it had a fish body and a snake tail. So that's the reason why we named this bacteria for Hugel plasma. And I'm going to introduce some about the bacteriophage, why it can help the host cell to degrade the nucleic acid. So for us, maybe you are familiar with the lytic bacteriophage because it can replicate within the cells, and then it can produce some of the sounds of the doctor's cells from the inside. But for the chronic bacteriophage, it is located outside the host cell genome. That is very like the plasmid. It is not located in the chromosome. It is located outside the host genome. This is we call that a chronic bacteriophage. So actually, in another DBC bacteria, we call that PVC bacteria, we also find this kind of things. You can see we use the polysaccharide as the substrate to do the enrichment. We can get this PVC bacteria. And next, we want to explore why this guy can degrade and then utilize polysaccharide. So we did this transcriptomic analysis. So after that, we found that actually a lot of genes associated with phage, like the phage tail protein, phage paw protein, the expression about the phage genes were evidently expressed. So it's very strange because we think maybe some genes about the polysaccharide degradation or transportation should be upregulated. Why the genes about the associated with the phage is upregulated? So maybe something about the phages associated with the degradation happened. So we did this purification about the virus from the supernatant of the cultivation. So you can see we got this filament phages from the cultivation cells. And we observe that it's totally filament. It's very long. It's about, you can see the bar is 1 micrometers. And also for this bacteriophage, it is named a virus, but it cannot kill the host cells. So you can see these filament phages can go inside and come out from the cells, but it will not kill the cells. And also we sequence this virus. You can see for the red arrows, it is genes about the polysaccharide transport and the degradation associated proteins. And also for another phages also contains several genes associated with the polysaccharide degradation. So it demonstrated again for this model, like for the host cells, it wants to use the polysaccharide, but for some reason in the genome, it is short of some essential genes associated with the polysaccharide degradation. So for the phage, they can encode these kind of important proteins. So they will help the host cell to use the polysaccharide. And also the host cell, it will provide some space for this virus. So it build this very, very friendly interaction actually. So it's very special. I think maybe in the deep sea, there are a lot of this deep sea bacteriophages. Actually, they will not kill the host cell, but help the host cells to utilize some polysaccharide or nucleic acid or other things. So just like in this paper we just published last year, so we show that actually in the deep sea there are a lot of very, very novel virus, or we call that bacteriophages in the deep sea. So they can help the host cells to degrade the polysaccharide, nucleic acid, protein. So maybe in the future, if you want to do something interesting about the bacteriophage, don't only focus on the lytic bacteriophages. You can expand your research areas to maybe this, we call that chronic bacteriophages. This is the first sample. And this is the second sample. We also use a polysaccharide to cultivate some archaea. We call that this methane production archaea from the deep sea. You can see it produces a lot of methane from the tube. You can see the bubble. It's bubble column. It produces a lot of methane. So it's very special because it can use this pectin to produce the methane. This is very, very novel pathway because right now it has 5 different pathways for production of methane. So actually we found this 6th one. That means it can use very complex matter like the polysaccharide to produce the medicine. So actually for this one, we will give more details in the future. And for the third example, I can show you some very interesting deep sea bacteria. We use the alkene as a substrate to do this enrichment. You can see we culture this very interesting morphology deep sea bacteria. It has an arm, a long arm, and its head. For the growth or for the cell division, you can see it has a budding, and the budding grows more, and more. When these two different parts becomes almost equal, it divides to different parts. You can see it has an interesting morphology, and also it has a very special self-division manner. And also, if we cut this deep sea bacteria into different slices, you can see this is the head. It contains the cytoplasm. And also this stalk, we call that arm, it is totally empty. From the stalk to the cytoplasm, it can transport the nutrient into the different bubbles. We call that maybe vesicle. So we did this observation in details. You can see we labeled this cell membrane with the red one. This is the cell membrane. And also we did this label for the hexadecan with the green color. You can see if it's merged, you can see the green color was transported to the for the cytoplasm for utilization. It's very interesting. And also, if I labeled the hexadecan with the FITC, you can see it's between it becomes a green color. So you can see it can go inside the cell cytoplasm very quick. So we did this photo every minute, and I made this video for about 1 hour picture taken. So you can see the video records the metabolization for 1 hour. You can see it go into the cell cytoplasm very quick. So it's very interesting for this guy. And also, if we cut the cell into different parts, you can see the different phase for this cell division. You can see for the panel A is only one cell, and then become longer and longer for this panel C, panel D, panel E, panel F. So for the panel F, you can see clear for the cell division part between two different parts. And then for the panel J, panel H, it finished the cell division. It become two different cells. So you can see the totally whole cell division and growth part. So why this guy has this very special morphology? So actually, we got this sample from the deep sea substantiation reaction environment. So you know, for this reaction, it can produce a lot of organic matter. So for this guy, they want to utilize, or they want to get this organic matter from the seawater. So it has to evolve this very interesting morphology. It has become longer and longer. Only become this very longer morphology, it has more chances to get this organic matter. Just like the giraffe, it only has a long neck one, it can survive. Otherwise, it would die. So that's the reason why they have these two long stalks. Okay, so for the third part, I will go introduce something about the utilization of light. So this is the deep ocean in the mind of most people, right? This is totally dark. You think maybe in the deep sea, it's totally dark. Actually, it's totally different. Actually, this is the real deep sea. You can see for the deep sea hydrothermal vent, it has a geological light. It is kind of this light. And also for other animals like the fish or other germs, other things, you can see it has a lot of light. So next question we want to ask, have deep sea microorganisms evolved special ways to use the light energy? So actually it is the answer is yes. So you can see we got some deep sea cold sea bacterium. It can use the blue light and sense the blue light and utilize the blue light by the blue F dependent pathway. This is use the blue light. And also we found another deep sea bacteria. This strain conducts a typical phototrophic lifestyle in both laboratory and deep sea condition. And also, we got this deep sea bacteria. It can use blue light. You can see the possibly it's very complex. So, and finally, they can use the blue light to produce the sulfur and to store this energy in the sulfur element. So the long goal, I think we will concern something about, we want to address two questions. The first one, what is the diversity and the broad distribution of light utilization microbiome in deep sea? And then the second one, whether photosynthetic life derives from the deep sea environment. For the first part, I want to tell you some even interesting things about the production of dark oxygen. So this picture, you can see it shows the cold ship and the deep sea hydrothermal vents. For the cold ship one, you can see a lot of animals. The white one is shrimp. The brown one is the oyster. And for the hydrothermal vents, you can see even more. This is the shrimp, the white ones. You can see we calculated one time for 1 square meter, it has 3,000 shrimp. So it has so small space. Why? It has so many shrimp there. So you can see actually for this deep sea hydrothermal vent, it is short of oxygen. You know, for the animals, it totally needs the oxygen to breathe, right? So actually from the center of this hydrothermal vent, the oxygen concentration near to 0 mg per liter. And even far from 3 liters, it has only 1.5. Actually, for most of the animals, they need 3 mg per liter to breathe. So next question, how do deep sea animals to get oxygen for breath? So that is a very obvious question, right? So actually for a couple of years, there is a paper they published, they found that actually for the deep sea, they have some enriched, they have some the oxygen. You can see this green curve shows the concentration of the deep sea. It becomes higher and higher with the depth of the deep sea. But for another one, so you can see the concentration is very closely related to the concentration of nitrate. So what's the reason? What's the reason why the concentration of the deep sea oxygen is closely related to the nitrate? So right now, we know that the deep sea oxygen, most of the scientists think maybe they come from the thermal thermohaline circulation. That means the deep sea water, the formation, and then they will transfer this oxygen to other deep sea. So we want to ask whether circulation is the only source of deep sea oxygen. So actually now they have some papers they published. So in addition to the oxygen produced by the photosynthesis, they also have some example for the dark oxygen production. So what is dark oxygen? So that means it is not dependent for the photosynthesis. So right now they have some different pathways. The first one is chloride dismutation. So you can see it produces oxygen from this chemical. And for other very typical pathway, they call that nitric oxide dismutation. For the first one, it becomes nitrite to the oxygen. For another one, also is very similar. We call that dark oxygen. So one time when we do the cultivation from the deep sea hydrothermal vent, so we found a very special deep sea bacteria. You can say when we culture the deep sea bacteria in the anaerobic sense, we always put this chemical in the medium. So that means if it has oxygen production, so this guy will transfer the color from the very colorless one to the red one. So it means some oxygen was produced. So when we do this cultivation, you can see from the 12 hours, it turns a little bit pink. For the 24 hours, it's totally pink. And for the 48 hours, it becomes totally red. So that means it produces some oxygen. because we know that it cannot do the photosynthesis, maybe in the indicate that it produces dark oxygen. So we check this medium. Yes, it produces a lot of oxygen. You can see the concentration of the oxygen about 70 or even 100 micromolar. It is very high. And we check this oxygen. You can see we transfer this oxygen from the bottle to another tube. You can see it can support the growth of the ants, and also it can support the growth of the locust. That means that's a real oxygen because uh, compared to the control, it can support the ants or the locust even longer. So maybe couple hours or even 10 hours for the longer growth. That means this bacteria can produce a lot of oxygen. So what's the mechanism? So we did a lot of very good jobs. So finally, we know the conclusion is they produce the oxygen for the nitrite, but not the nitrate. For the nitrate, but not the nitrite. You can see it can. We label this oxygen, and then eventually we found this free oxygen was derived from the nitrate. So what's the function for this oxygen? So in the deep sea, we know it has a lot of mineral. So we call that manganese oxide. So you can say for the formation of this guy, in the help of the microbe, it needs the oxygen. So if this bacteria can produce the oxygen, maybe it can help to form the manganese oxide. So what's the answer? It is yes. You can say when we culture this deep sea bacteria together with this element, you can see it can form this mineral. And if we check that, it is yes, that is manganese noodles. And also, this is the real the manganese noodles from the deep sea. And this is the formation of the manganese noodles formed by our deep sea bacteria. You can see it's very similar. It's the formation and the morphology is very similar. That means actually in the laboratory condition, we can use this deep sea bacteria to form the real manganese noodles. So what's the ecology function of this dark oxygen? So we did a very special test. You can see in this bottle, we put this tube. It's a half. It is open in the upside. So you can see we culture this totally anaerobic bacteria, archaea, in this side. If this guy produce oxygen, it will be released and then go into this side. And this anaerobic microbes cannot grow. The answer is yes. You can see for the control one, it has very better growth, but for the production of the oxygen, this archaea, because it's a totally anaerobic ones, it cannot grow. It cannot grow. And for another thing, so you can see it produces the oxygen. It can support the growth of the aerobic microbes. We did the same thing. So that means yes. So for the dark oxygen, it can produce a lot of ecology function in the deep sea. So for this whole overview, you can say if this guy can produce the dark oxygen for some reason, and then it can support the growth of the faunal community from the deep sea, and also it can help to formation of these nodules, and also they can support the growth of the deep sea aerobic animals of the aerobic bacteria or archaea, etc. Actually, it has a very, very important effects in the deep sea. So actually, in the whole deep sea, it has more mysterious process inside. So because the time is limitation, so maybe in the near future, I can tell you more interesting results in our lab for the cultivation and for the deep sea process. And for the last part, I maybe for some 10 minutes, I can show you something about the plastic degradation by marine microorganisms, because in the first introduction from your side, you mentioned that you will find some maybe bacterial enzyme from the deep sea or from the ocean. So I will show you some plastic degradation results from our side. So this is a very typical plastic degradation from our side. You can see we found a marine fungus. It can has a very good growth in the top of the plastic. We use the PE. The PE is very difficult to degrade. So you can see compared to the control, it can colonize on the top, on the surface of this PE plastic very well. And also when we remove this fungus biomass from the plastic, you can see it produces a lot of deep holes. So that means something happened. But the time is very slow. You can see if they produce this deep hole, it need about 4 months. It's very long. So we did some optimization. You can see after our optimization, it need only 3 weeks can produce thousands of the deep holes. And also not for the PE, for the PP, for the PR plastic, it also produce the same thing. You can see for the deep holes, a lot of. It's very obvious. So what happened? So you can see for the PR, it's a little bit easier to degrade. So you can see for the PR, for the first day, for the 1 day, you can see the film already be penetrated from the bottom. And for the 2 weeks, you can see the PR film already degraded to different parts, and also the color turned yellow, dark, and also turned different pieces. So we did this video for you. You can see for the first week, second week, 4 weeks, it totally degraded, and also the color totally dark and different pieces. You can see for the PR, it's very obvious. And also we explore some single enzyme. We call that curtinase from this fungus. You can see for the PBAT plastic, it only needed 24 hours to degrade to different parts, and also degrade this PBAT film to monomer. And also for the PCL, it's more obvious. You can see for the emulsions, 1 minute. 1 minute it can totally clear. And now for the even for the film, it only need 2 hours to totally degrade, and also to become this chemical to monomer. It's very obvious. So in the long go, maybe we will develop some more enzymes to do the degradation for more plastics. So for the long go, we want to build this cyclic economy. Why? Because when we did this plastic degradation, it produced a lot of the fungal biomass. It is also a bio waste. So how to do that? So because we know this fungus, it can produce a lot of metabolites. So for this metabolites, maybe it can develop to the biomedics, and then for the drugs. So and eventually we can build this circular economy. And also for this fungus, yes, it can produce the biomedics. You can see it can inhibit the MASA, and for Bacillus vibrio. That means this plastic degradation fungus can produce the antibiotics. And also for this MASA, you can see it can produce the inhibition effect even better than the vancomycin. So maybe in the future, we can use this guy to develop the antibiotics. And also, it is very safe for the zebrafish, and also for the human cells. That means in the near future, we can use the fungal biomass to produce this safe biomedics in the antibiotics in the future. And also, we can build this real cyclic economy. Like if we do this degradation for the enzyme, so we can build this microbial cell factory and to produce the bioproducts eventually. And then we will use biomass, we can produce the antibiotics, and then to make this drug, eventually we can build this circular economy. But it is a very long goal, and also very difficult to fulfill this goal. But maybe in the future, we need different measures, different like the chemistry, biology, and the different governments, different countries for this very, very difficult task in the long goal. And this is the last slide for my presentation. So just like I mentioned, actually, I noticed that actually Dr. David told me for your side and our institute, we already built this connection maybe 1 or 2 years ago. I forgot. So you can say this is records. Maybe you know some familiar faces in your side. This is our head is of this institute, and we signed the document. That means we already the friend a couple years ago. And also, just I mentioned, I also had some funding from the ANSO, ANSO, already built some connection from your country and the Philippines and other countries from Asia. So maybe in the long go, we will do more collaboration for the deep sea microbiology and also for the plastic degradation on the enzyme foundation, other things, I think. So lastly, so I would say thank you very much for giving me this chance to do my presentation, and thanks to all the members from our lab, and also thanks for your attention. Thank you.
Okay, thank you very much, Professor Sun, for very insightful and inspiring presentation. Professor Shun, I heard you want to introduce your lab to us. It is better to start now or after question and answer session?
I think maybe after the discussion. So if
Okay,
you have some question, we can discuss. After that, I can show you some
okay,
the overview of our lab.
okay, okay. Thank you so much. So yeah, it is very interesting presentation because I also working with the fungus and bacteria degrading plastic degrading bacteria and fungi and also enzyme. And in your work, you very uh, inspiring results. Yeah, like you found the dark oxygen and also you convert the plastic to become antibiotic and become the circular economy. So for the participant,
Yes.
if you have any question, please feel free to write in the chat box or raise hand a picture and kindly mention your name and please keep your question concise so that we can accommodate as many as participants. Yeah. Okay, please. Okay, we have a question from Bu Sandy in the chat box. Uh, I will read the question option. In your experience, is it more appropriate to begin microbial cultivation using sterilized natural seawater in order to preserve unknown growth factor and trace element that may be essential for uncultured microorganism? Alternatively, could artificial seawater be used effectively for this purpose? And would you recommend its use at the initial state of cultivation? So kindly your response, obsession.
Uh, okay. Uh, so because the voice is not very clear, so if I didn't say clear, so you can remind me. So
Okay.
actually, for the bacteria or the archaea isolation from the deep sea, it's kind of it's tricky actually. So we call
Mm,
that deep sea difficult to culture. You can see it's very evident because it's very difficult to culture. It has two reasons. One is, it's very difficult to get the sample from the deep sea, like the deep sea cold sea, the deep sea hydrothermal vents, or even the trench, Mariana Trench. So it's very difficult to get this sample. It's very valuable. So if you don't have this ship, the big ship, you cannot go there. So the second one is if you get the sample, so because just like I mentioned, so because a lot of the deep sea microorganisms, they are totally anaerobic. So if you get the sample and then put on the board of the ship, it is aerobic. So most of the microorganisms they died. So that this is the third reason, and the last one I think maybe most of the lab they are working on the aerobic things, but for the anaerobic So we are very short of the experience. I think maybe in the near future, we can talk about the details. And also, like you mentioned in your side, maybe you have some difficulties to get the sample from the deep sea. Also, Dr. David mentioned that. So maybe in the near future, because we have this collaboration, maybe we can have some shared samples. I mean, the strategies. So maybe we can go to your side. You can join us for some calls maybe in the near future. So you can get the valuable samples and we can collaborate together to do this cultivation.
Okay, thank you, Professor Shun, for your helpfulness, and thank you also. Question from Sandy. Sandy, do you want to open it?
Enough of, yeah, it's clear.
Okay, so we open next question. If you have any question or comment from the audience, please. You can use open mic. Okay, for Professor Shun, I want to ask about the dark oxygen. It is very
Okay.
interesting. You found the microbe produce the dark oxygen, and uh, for your perspective, how significant this phenomenon in shaping our understanding of the sea microbial ecosystem? I mean, and also what the implication might it have for the future research on the difficult to culture microorganism? I see in your slide that this dark oxygen is enhance the, I mean, the supporting the growth of the aerobic bacteria, and also can inhibit the anaerobic bacteria. So what is the difference between the oxygen we usually found in the light condition and with oxygen in the
Dark
dark
oxygen.
ecosystem? Yeah, yeah. Do you can explain this? Thank you.
Okay. Yeah. So actually, for most of the absolutely anaerobic deep sea microorganism, they don't like the oxygen.
Yeah.
So that is absolutely anaerobic. So like the methodologic are here, they are afraid of the even you have very very little oxygen inside the medium, they cannot grow. So, but for the production of dark oxygen microorganism, so they can produce some oxygen, but they cannot produce so much oxygen because in the real deep sea environment, if they produce the dark oxygen, they can be released to the environment. So actually, because they produce the dark oxygen only because they want to use the nitrate for the substrate. So actually, the oxygen only something they produced by this process.
Yes.
Actually, so they can use a little bit the oxygen for the for the growth or something, but they don't like so much oxygen because you can see in the bottles when we culture this deep sea bacteria, they produce about 100 micromolar. But if they produce more, they will stop this production because they don't like so much. Because just like I mentioned, in the real deep sea environment, if they produce more oxygen, they can release to the other environment. So that is not too much inside the growth environment. So that's look like the contradict, but actually for most of the deep sea anaerobic microorganism, they don't like the oxygen. But for this one, it's special.
Okay. Okay. Thank you so much, Professor Shun. Okay, Bu Dewi Zilda, please.
Yeah. Thank you, Professor Son. It's a very amazing presentation. I think one slide to another slide is very interesting. Then sometimes maybe we didn't realize that we already at the end of the presentation. So very amazing.
Yeah.
So I want to ask about something. I saw in your presentation about the microorganism related with polysaccharides. It looked like
Yes.
that. Is it a produce enzyme who degrade polysaccharide and then degrade the polysaccharide in the environment, or they consume polysaccharide and then produce oligomer or something like that? Is it related with the enzyme polysaccharides?
Okay.
Okay,
So
so
I show you this slides. Uh, yes, actually, we found some many deep sea microorganisms. They use the polysaccharide to for the supporting of the growth. Actually, in the a lot of deep sea environment, we found that it has a lot of different polysaccharide, like the chitin, the lamivudine, and so we just like I mentioned, actually for the degradation of the polysaccharide for the deep sea microorganism, we didn't find some special enzyme. Actually, for the degradation and utilization of polysaccharide, it's very complex. It's not by the single enzyme, just like this model. So you can see actually this deep sea virus help the host cell to utilize the polysaccharide. So we found some very special proteins related to the polysaccharide degradation in the genome of the virus, but not in the host cells. So that means just like this model, actually it is the virus to some extent help the host cell to utilize the polysaccharide. So actually, we still didn't find any single enzyme can do this very complex degradation. So that, Dr. David, that's my answer.
Okay, thank you.
Okay, thank you, Bob. Okay, we have a question from Pak Yes, this is our previous deputy. Prof. Okay, yeah. Do you want to open mic?
I think I
I
have
think I
already
have already
written
written.
in
Oh yeah, okay,
my.
Prof. Okay.
Okay.
Thank you for
Thank you
joining
for joining this
webinar, Prof.
webinar, Prof.
Okay,
Okay,
Prof. Sun,
Prof.
how
Sun.
did you collect the deep samples? First question. And the second is, did you use like a pressure retaining samples, or your sample experience decompression during retrieval? How important mimicking the ecological condition of this of deeps, such as high pressure, low temperature, and high temperature during cultivation? Thank you.
Okay. Yeah. So actually, for a lot of scientists, they if they don't real do the experiments or the something about the deep sea, so they just worry about the pressure actually. So actually, for most of the deep sea microorganism, the pressure is not the big issue. It's not the biggest issue actually. So the reason because most of the deep sea microorganism, they can endure the pressure change between maybe the 1 atm to 100 or even 1000 atm actually. So that means they can survive in the regular pressure, and also they can survive in the very high pressure. So the pressure thing is not the biggest problem. Actually, for us, we found that the biggest issue is oxygen. It's oxygen. So actually, for the pressure, in the deep sea, sometimes they have this circulation. Some bacteria, they can be maybe in 1 minute they can in 1,000 meters. Maybe in another minute they can go to 2,000. So they can change their pressure inside to cope with the outside pressure. It's very quick. It's very easy. So for us, I think if you want to get the real good sample, you need to very to notice about this the oxygen thing, but not the pressure thing. Okay.
Thank you.
Okay, thank you, Prof. Shun. So we're still open some question. If you have any question from the audience. Okay, so Proption, I'm also working with the PTase enzyme and also glutinase and lactase enzyme. So I want to ask you about from the fungi you isolated from the deep sea. So you present about the glutinase. First question is this glutinase enzyme like new or same with the other glutinase? The second question is, do you found any enzyme like lactase or PTase or carboxylesterase enzyme that also responsible for plastic degradation in from the deep sea microbe?
Okay, so actually from this fungus, we found that this cutinase, we call that CART10 cutinase. So it's not very novel actually. It's from this fungus, but it's very special because for the plastic degradation, normally right now, the enzyme, if they can work well, you need the high temperature, right? So maybe 70 or 75 degrees. But for this one, because they collect from the ocean environment, it can work very well in the room temperature, like 25 or 28 degrees. So I think it's much better because for the long goal, if you want to do the industry application, you cannot do this degradation in so high temperature because you need to put a lot of money to produce high temperature, maybe 70 or 75 degree. But for us, we can do this degradation in the room temperature anywhere. So I think this the it's very I think it's better news for us. For another sense, for this fungal strain, it has a lot of different enzymes. Actually, for us right now, I think maybe we just find a corner of these enzymes. Maybe in the near future, like you mentioned, it has I think maybe 100 or 20 or even more, 200 potential enzymes inside the cells. But sometimes we just worry about maybe one single enzyme cannot do so much complex task. So we need to use this kind of combination, maybe one or two or even three or even more enzymes together to do this, especially for the PE degradation. I think it cannot be performed by only one single enzyme, maybe couple of enzymes in the near future, but it's a very long goal.
Okay, thank you, Professor. I hope that we can collaborate in the future for this plastic degradation.
Okay.
Bu Devizilda, do you have any additional question?
Yes, from your question, Pak Dede, Professor Sun, you mean that it's better for us to use the bacteria directly to the substrates instead to work to find enzyme purify or something like that? It's better we use the bacteria directly, like to degrade the plastic or something.
In case of we use the fungus.
Use the fungus, not the bacteria.
Okay. So actually, we have that problem a lot. Like when we use the bacteria, for example, for protease, we use the bacteria, and then they can degrade like keratin. But then we try to isolate the protease. Keratinase is one of the protease. But then we try to isolate the gene and then overexpression or something. But they cannot work better if we use the bacteria directly. Yes.
Yeah. So actually the same thing happened for us for this fungus, the fungal enzyme. For example, for this cutinase, when they overexpressed in the E. coli cells, the expression is pretty good. The yield is high, but it cannot work. It totally cannot cut the plastic. So we have to transfer this hot cell to the yeast. So when we overexpress do this purification from the yeast, so it can work very well. So that means you in one you counter some bacteria, it can it can degrade the cellulose or some other complex substrate. So when you try to overexpress one single enzyme in the E. coli cells, maybe it cannot fold well. I mean, even you can see the expression is good, but maybe the folding is not correct. So you need to do more tests. Maybe you need to change the expression cells. Maybe you can try the yeast cells or other cells, I think. So, but it's a very complex, just like I mentioned, for the cellulose or plastic, it's the structure is very complicated. Maybe one enzyme cannot work well. You need to try the different combination. That's my suggestion.
Okay, thank you, Professor Shun.
Okay, thank you, Professor Shun. We still open the question if you have any question from the audience. So actually I have still a question. So Prof. Shun, in your experience, you already have successfully isolate and culture the many deep sea microbes, which is we know as a difficult to culture microbes. And what do you think about the application? I mean, if we try to find a new application of the deep sea microbes, should we try from the like ecological or physiological function of this deep sea water, deep sea microbe in the deep sea, or we can like elevate the, I mean, the different function. If we can culture the microbe and then we can use this microbe for any application, or it is specifically only like
Yeah.
what they usually have a function in the deep sea.
Okay, yeah. So for example, I can give you two examples for the for the deep sea methanogenic archaea. It can produce a lot of methane. So actually, for this strain, it can produce much more than the strains I already saw in other area. So actually, for when we do the cultivation in the bottle, because it's an anaerobic one, so maybe from time to time, after a couple of days, we need to release the methane from the bottle. Otherwise, it will boom. So because it produces a lot of the methane, in China, not like a country, because in China, we have a half of our people, they live in the very north of China. In the winter, it's very cold. So in the long goal, we want to use this methane for this in the winter. They can produce a lot of warmer environment. So and also it can totally to decrease the cost. So this is one example. Another example for the dark oxygen production for this bacteria, so in the long goal, we even imagine to use this guy to do some things in outside our Earth. Because when we try to move to the moon or other planets, so the human totally need the oxygen. So if we can put this guy to the moon or other, yes, they can produce the oxygen, and then we will change the environment better and better. After that, our human being can transfer to there.
Yes.
And also, so for the application, we know some bacteria in our human inside, so they are totally anaerobic. Some pathogen, they are totally anaerobic, but they are very very dangerous for us. So for the some bacteria in the stomach, right? So if we can develop this dark oxygen produced bacteria to our human beings, you can transfer this guy to the inside of our bodies. They will produce some oxygen and they will inhibit the growth. And also for the cancer cells, for the cancer cells, if they want to grow faster, they don't like the oxygen. So
Yeah.
if you do some tissue culture since because we need to put some dioxide,
Yeah.
CO2 into
To make
the
the
culture environment. Yeah. So they don't like the oxygen. So we want to produce oxygen, and then they can inhibit the growth of the cancer cells. So I think this is two samples. It looks like it's long, but I think maybe
yes.
if we can do some better job, maybe in the near future we can fulfill this very good
Yes.
picture. Yeah.
Which is very interesting and very yeah, interesting topic research research topic in
Okay.
the future. Yeah. Thank you so much, Professor Sud, for your thoughtful response, and also thank you for all the participants for the engaging the questions. So
Okay.
I think
So I think
would it?
we need to see the
Yeah.
video or
The
something. Okay.
okay.
Yeah,
The
okay.
laboratory,
Okay.
right?
Proxon, if possible, can we see your laboratory facility
Okay.
by using?
So yeah, I
Thank you
can
so much.
show you a little
Yeah.
bit. So
Okay.
maybe
Thank you
in
so much.
the next time, if you have a chance, you can come here. I can show
Yes.
you in the real environment, and so you can see and how can we do the arrangement for our lab. So I can show you around.
Okay. Thank you
So
so much. Yeah.
yeah, this is
What
my
city your lab, Professor Shen?
sorry, what
What city in China? Which
Qingdao.
province? Qingdao. Okay.
Yeah, yeah, yeah. So this is my office. You can see a lot of green things in the wall of
Oh.
my office, and so I will show you a little bit the. So this is the lobby, and this is the door of my lab, and it's a whole view my lab. So you can see, and in this lab we have all the equipments, all the bench work. So you can see, and this is for the autoclave, and this is the bench, and also you can see a lot of plates, scenes. So this is the half of my lab. You can see the shaker,
Mm.
and this is for the gel sense electrophoresis, and also we have this container for the chemicals. Yeah, this is the half of my lab. You can see, and I'll show you this very special the cultivation equipment. So for this one, it is 4 degree about the inside these cells in these containers, and then we can do a very long growth. So maybe 1 year or 2 year. So maybe next time when you come here, I can show you the details.
What is use what why you use the pump there? Oh.
So basically for this column, you can see, so you can see a lot of sponge inside the column.
Oh, okay.
So for one sponge, it contains one different sample. So you can did this growth or isolation or enrichment. You can say, so in one column, maybe you can do maybe 20 or even more, the
Oh,
different cultivation. And for this column, the good thing is from the top side, you can put some new medium there, and then from the bottom of this column, so the waste they can come out. So for this one, it's it can always keep the fresh things, because sometimes the fungus or the bacteria, they can produce the metabolites inside and then inhibit the growth. So that means you can put the fresh medium from the top, and then the waste can come out from the bottom. So and also you can do a very long, because this is 4 degree, it totally mimic the temperature from the deep sea, 4 degree or 2 degree, and then you can do this enrichment for 1 year or even 2 years.
Okay. Interesting.
Okay. Yeah.
It is very new for me.
Yeah.
In 1 year, we have to give an output, so we cannot wait for 2 years to start the experiment and then get the outputs. But in
Yeah.
Professor Sun laboratory, they can run that equipment from the beginning and get the result maybe 2 or 3 years later.
Okay.
Yes.
Okay.
Yes. Yeah. This is the another part, another half of our lab. You can see, so this is HPLC because, like I mentioned, we did from the very beginning part, from the cultivation,
Mm.
the study about the live process, and also the metabolites. So we use HPLC, and this is FPLC. We did the purification for the plastic degradation enzyme, and also the microscope, so we can do the tissue cells or other things. So I think, like Dr. Debbie mentioned, we can do it very from beginning to the very bottom. So we can do all the things in our lab. So this is the microscope, and also this guy for the is for the Western blot or other things. Okay, so this is the centrifuge, and also our students are doing the the experiments. So you can see we are preparing some tubes. So we did the anaerobic cultivation by this kind of tube. So this small tube, you can see the black lid. So for this guy, it is totally anaerobic. So
Oh.
yeah. Okay. So you can
Professor
see.
Shun, do you have like a student also from the university, or
Sorry, what?
do you have also like a doctoral student in your lab?
Yes. Yeah.
Okay.
So you can see we have this kind of tubes a lot. So because I can show you even more. So we have this this hood is for the we call that a glove box. So it can do something about the anaerobic purification or isolation in this box. And also I show you we have a lot of high temperature incubators. So you can see we have this 1, 2, 3, 4, 5. We have 6 actually. So you can see the temperature is, we set the 60
16?
degree,
Yes.
70 degree. So you
70.
will see the amazing things inside. Okay, you can see, right?
So many, so crowded.
Yeah. So yeah, a lot of tubes, a lot of the you can see the bottles. So the black thing is the the alkene. So we use
Mm.
this oil for the substrate for the cultivation, and also, so you can see a little bit of the anaerobic. The growth is not so well, but actually for the anaerobic bacteria, it already very well. So you can
Mm.
see. So it's a little bit cloudy, right?
Mm.
So so we have 6 or 7 incubators. Every one you can see totally full. Yeah, with this you can see this for the enzyme
That's
screening.
okay.
Yeah,
Oh.
it's yeah. So I think you did a similar things. So because this the high temperature, you can see 70. So that means the enzyme is a high temperature, so it can
Hemophilic
do the
enzyme.
maybe 70 degrees something. And for this one, so you can see it's a little bit different because we
Dark.
put this, because uh, even I mentioned some deep sea microorganisms, they like the light, but they don't like very strong light. They like the very faint light. So that's the reason why put this guy here. So that means it can prevent the light, very strong light. Yeah, we have kind of this things, and this is the picture I show you. We did so you can see the degradation.
Plastic. Oh,
Yeah, plastic degradation. You can see.
polyurethane.
This is the PUR.
Yeah,
PUR,
polyurethane
yeah.
foam.
Yeah, and also we did this kind of sponge degradation sponge.
Yeah,
Yeah.
yeah.
Yeah. So yeah, like I mentioned, and also this is the zebrafish.
To see the detoxification, no?
Yeah, because we want to find some enzyme like the protease. It can cleave the protein to different parts, maybe three peptide. This kind of peptide, they have very useful area for the health sense. So we can test their effects in the zebrafish. So that means you can see we can did all the things in our lab. And
You have so
also we have
many a big topic.
yes, yeah, and also we have this food. It's a little bit special because we can did the tissue cells here.
Okay.
I showed that we can did the test for the human cells or even the cancer cells. So this hood is a little bit different. So that means it can only do the test for the tissue house, but not for the bacteria or fungi. Yeah.
Okay.
So and also for this is extracts of the chemicals or the metabolites from the deep sea microorganism.
Yeah.
So yeah. So I think that's most of the things in my lab. And also we have I show you a little bit. Because we are the Institute of Oceanology. So you can see just our outside our window, you can see the seaside. Can you see the?
Okay, yeah.
Yeah, it's very. Yeah, you can see, right?
Yeah. It is a
Yeah.
sea?
Yeah, that's the sea.
Okay.
Yeah.
That's
The beach.
yeah, that's
that's
we call that Huanghai in China.
Oh.
Yeah.
So for Sun, I saw in your presentation, you also make a reconstruction of the mitochondria structure. Do you use like a cryo-EM for this 3D structure?
Sorry, I missed your question.
In your slide, I saw you reconstruct like a mitochondria in the green
Okay,
one.
okay.
So how you reconstruct this one using the TEM?
Yeah, we use the TM, for the one cell, we can cut into different pieces, and then we do this reconstruction. You can this green one, we call that a Kimber sense.
Yeah.
Because we can do this reconstruction, we can reconstruct the real structure inside of the cells.
Yes.
So and
So
also
you
the interesting thing, we also find some mitochondrial protein in the genome.
So you also have like a TM machine in your lab,
Yes.
in your academy?
In not in my lab, but in our institute, we have all the TM, SEM, or even better. Yeah.
Interesting.
Okay. Yeah, I think that's most things in our lab, but not in very details. I mean, maybe in the near future you can come
Yes.
here together with Dr. Davy, and I can show you more details in my lab, in my institute, and maybe I can show you the big ship in our lab, and also the robot. It can dive to the deep sea. I mean, it can do the sample collection or other things for us.
Okay. Thank you so much,
Okay.
Professor Sun, for a virtual visitation in your laboratory. And I think
Okay.
it is very valuable knowledge for us and also insight for us today. And we hope that we can like make a very good collaboration in the future, especially can visit your laboratory in the future.
Okay.
So I also would like to thank all the participants for your active participation. Now I will hand the session back to the MC. And again, thank you very much, Professor Shun, for your presentation, for your sharing, and also for a virtual visitation to your laboratory. Thank you
Okay.
so
Thank
much.
you. Thank you very much. Okay.
Okay. Okay. Thank you, Dr. Dede, and thank you, Professor Chominson, for your amazing presentation and also interesting laboratory tour.
Okay. Thank
I
you.
appreciate your fruitful knowledge in this webinar, and the Research Center for Applied Microbiology will give you appreciation as an honor certificate as a speaker in today's webinar.
Okay. Thank you.
You're welcome. And
Okay. Thank you.
for the next session, we arrive in end of a webinar. Hope this webinar bring us the fruitful knowledge about deep sea microbes and how to manage the microbes and how to explore the value and advantage for those deep sea microbes. Big thanks for Dr. Ahmad Fatoni, Professor Chow Minson, and Dr. Dede, and to all the participants. See you on the next webinar.
Okay.
Okay.
Thank
See
you.
you.
Okay.
Thank you so much.
See you soon.
Professor Shun.
See you. Okay. See you.
Bye bye.
See you. Bye bye.
Bye bye.
再见，Professor.